A key element of the UK Spine's mission is to support development of drugs which target biological drivers of aging. Toward this goal, the UK Spine's Bridge program has funded the project Impacts of Mechanistic Target of Rapamycin Inhibition on Aged Human Muscle. The principal investigator is Philip Atherton, the Chair of Clinical, Metabolic, and Molecular Physiology at the University of Nottingham. Collaborators on this work are Daniel Wilkinson, Assistant Professor in the Faculty of Medicine and Health Sciences, Ken Smith, Professor of Metabolic Mass Spectroscopy, Bethan Phillips, Associate Professor of Clinical, Metabolic, and Molecular Physiology, and Matthew Piasecki, Assistant Professor of Clinical, Metabolic, and Molecular Physiology, all at the University of Nottingham, as well as Dr. Simon Wynne-Jones, a reader in musculoskeletal aging at the Institute of Inflammation and Aging at the University of Birmingham. Muscle is important for maintaining whole-body metabolic health. Reduced skeletal muscle mass and function are associated with increased cancer-related deaths, delayed post-operative recovery, and reduced independence, among other age-associated morbidities. Between middle age and age 80, approximately 30% of skeletal muscle mass is lost. Mechanistic target of rapamycin, or mTOR, is involved in the maintenance of healthy muscle. The diagram is from a 2018 Aging Research Reviews article. It depicts characteristics of mTOR-mediated, age-associated changes in skeletal muscle. Exercise is currently the only strategy for countering age-associated muscle loss, also known as sarcopenia. Alternative or supplemental intervention would help address this clinical need. mTOR becomes overactive with age, and evidence suggests that this hyperactivity plays a role in sarcopenia. Rapamycin acts on mTOR, and, in animal models, treatment with rapamycin improves skeletal muscle preservation. Toward identifying an alternative intervention for sarcopenia, the research team is conducting a small-scale clinical trial to assess the impact of rapamycin on muscle health in older human volunteers. This project interrogates the impact of rapamycin on muscle health in an aged cohort. It also explores the interaction between rapamycin and exercise. For both resting and exercise cohorts, goals are to determine the impacts on muscle mass via DXA and MRI, D3 creatine tracer, and ultrasound, to also determine the impacts on muscle function by looking at MVC-1RM for strength, muscle power measures, physical battery testing for performance, and muscle nerve crosstalk, and to assess impact on muscle metabolism by investigating muscle protein synthesis, muscle protein breakdown, and regulatory pathways of muscle protein turnover. For this clinical trial, ethics approvals are established prior to initiation, along with recruitment for 16 male volunteers aged 65 to 80. Volunteers are randomly assigned to either rapamycin inhibitor or placebo, and baseline body composition and muscle strength and function are assessed. A leg exercise regimen for only one leg will be initiated and repeated 3 times per week for 4 months. Bilateral leg muscle biopsies enable the rest-assigned leg to act as a control. Biopsies are taken at 0, 2, 3, and 4 months. Arterialized venous blood samples taken weekly for the first month and biweekly months 2 through 4. Clinical uses of rapamycin include preventing organ transplantation rejection and treatment for certain cancers. Rapamycin's status as an approved compound presents a potential opportunity for accelerated route to clinic via repurposing for sarcopenia. Separately, the outcomes may be leveraged by other researchers to accelerate their own drug discovery programs targeting the link between mTOR and biological aging. To that end, early interest has been expressed by an industry player active in developing new interventional drugs acting on mTOR to mitigate biological aging. In addition to anticipated publications and presentations, this project has yielded knowledge exchange outputs, which include a new collaboration between the University of Nottingham and the University of Birmingham. Early engagement has been established with a startup developing rapamycin analogs, or rapalogs, to explore a path forward with the results of this initial study. More broadly, interest in rapamycin's potential as an agent protective against biological aging is widely acknowledged. This work will contribute to the body of knowledge leveraged by researchers globally toward dissecting relevant biological pathways involving mTOR and developing new or repurposed interventions acting on these pathways. Drugs designed to mitigate aging would be protective against multiple morbidities via a single intervention. Our current understanding of biological mechanisms driving aging is incomplete. Professor Atherton offers his thoughts on the question, how will this project contribute to a better understanding of the biological mechanisms of aging?
This particular project centers around the notion that a pathway, namely the mTOR pathway, becomes hyperactive in skeletal muscle of people of older age. And it's thought that the hyperactivity of mTOR, whilst typically mTOR is associated with positivity and positive growth signals in muscle, it actually becomes a negative occurrence. Therefore, the project I'm proposing is to use a drug which is called rapamycin, which is a naturally occurring compound which inhibits the mTOR pathway. In order to try and determine whether or not suppressing mTOR activity in age muscle will lead to positive shifts in skeletal muscle metabolism, favoring preservation of muscle mass. Of course, this means that this project is effectively centered around trying to mitigate age-related sarcopenia.